Hi everyone. In this lecture, we're going to talk about selected material from chapter 8 of our text. In this chapter, we focus on movement. We will spend the majority of our time in this chapter talking about skeletal muscles, so let's first take a look at the physical structure of a skeletal muscle. What we know is that most skeletal muscles are attached to bone on each end, so each end of the muscle is attached to bone by tendons. And skeletal muscles really help us do a couple of different range of motion. So the first is flexion. So flexion just refers to the ability to move limbs inward towards your body. So if your arms are out away from your body, moving your arms back into your body is flexion. And then in turn, we also have something called extension. This is where we're moving limbs away from our body. We also say that skeletal muscles help us with things that are anti-gravity or anti-gravity movements. So if you think about standing up, gravity would be pulling you down like toward the ground. But skeletal muscles allow you to do things against the force of gravity, which is essentially holding your body up. Here we're looking at the structure of a skeletal muscle. We have two different types of muscle fibers, outer and inner. Here, if we take a look, this is our outer muscle fibers. And then below that, we have inner muscle fibers as well. These outer muscle fibers are called extrafusal muscle fibers. These are served by neurons that exist in the brainstem and in the spinal cord that have axons that project into the extrafusal muscle fiber. These are really responsible for initiating muscle contraction. These are heavily involved in voluntary movements, so intentional movements of the limbs, for example. And these extrafusal or outer muscle fibers contribute what we think to be about 55% of your total body mass. Then, of course, we have those inside muscle fibers. These are more serving involuntary movements. So these are specialized sense organs in particular that are served by sensory and motor neurons. So one in and one out. So a sensory neuron sending messages or signals in, afferent, in toward the system, and then back out, efferent, via a motor neuron. These are also referred to as gamma neurons. These allow your muscle spindles here on the inside to remain more taut, and they also allow the alpha neurons and the extrafusal muscle fibers to continue to fire and allow you to do the voluntary movements. But again, we think that the intrafusal muscle fibers are more related to involuntary movements. Now let's take a look at sensory feedback. When you are having neurons in these different muscle fibers firing, what is that actually doing? On the inside fibers, those intrafusal fibers, we refer to those as being stretch receptors or stretch sensitive. So they're really going to become more active when your muscle is being stretched or extended out. These are also referred to as just length detectors. So when your muscle is actually lengthening out, these neurons in these intrafusal fibers are going to become more active. In particular, the ones that are located in what we call the Golgi tendon organ, which we'll take a look at here in just a second. And these receptors code degree of stretch by rate of firing. So there is more stretch when there is more firing, or more accurately, there's more firing when there's more stretch in the muscle. Let's take a look at what that looks like. This is a diagram from your book, and here we're looking at different ways in which a limb, in this example an arm, can be moved or extended. And we're taking a look at the firing of different receptors in the muscle spindles to see, when do they fire, how do they fire, what do they respond to. So here we're taking a look at two different muscle fibers. So we have MS1, which is muscle spindle 1 or muscle spindle fiber 1. And then we have MS2. And then we also have the GTO, which is the Golgi tendon organ. So here we're looking at an example of a slow lowering of the arm. What we note is that the Golgi tendon organ doesn't change, and muscle spindle fiber 2 also does not change, but the first one does. You see that it increases in rate of firing. So this can tell us really how these different receptors are responding to this type of movement and how this movement is made possible by the sending of these signals here. But when we do a more rapid or abrupt movement, what happens here is we also see an increased rate of firing here in MS2. MS1 remains largely the same throughout, but we see this increase in firing here as the arm is dropped. And this is really to signal there's a rapid change to muscle length. If you drop your arm rapidly, that muscle there is going to lengthen out quite a bit. And so MS2 is going to fire in response to that. And then finally here at the bottom, we have an instance where we're dropping a weight into a hand here. So what we see here is finally we see some responding of the Golgi tendon organ. And this organ tends to respond to proportion of stress on the muscle. So as weight is added to the hand, there's more stress or tension in the muscle here. So the muscle has to tense up, tighten up in order to hold this position while the weight is in the hand. So what we see here again is kind of an increase of firing because there is a sense that that muscle is being stressed. So what you can take away from this is that we have different neurons or receptors that respond to certain movements of a limb, in this example, an arm. Some are responding more to a quick lengthening of the arm, whereas the GTO, or that Golgi tendon organ, is responding more to just overall stress on the muscle. Now let's look at control of movement itself. So what happens when we actually move a limb and how that's controlled? First, let's take a look at reflexive control. So this is more of a automatic response. With reflexive control, what happens here is that the muscle is contracting very quickly when stretched. So there are a couple of different components involved that make this possible. We have a sensory neuron, so that's going to be the neuron that is detecting some stimulus in the environment. And then we have the motor neuron, which is going to allow us to actually move the muscle in response to that stimulus that's being picked up by the sensory neuron. And then, of course, we have the synapse between the two. In particular, first, we want to look at what is called monosynaptic stretch. So what that is that there's only a single synapse here, mono. A good example would be when you go to the doctor and they check your kneecap for a reflexive leg reaction. So they tap your kneecap to see if your leg will kind of kick out or jerk. This is a very interesting response in that this particular response doesn't involve the brain at all. That's why it happens so automatically. It's very quick. If we take a look at the pathway here, there is again only one synapse, again, hence monosynaptic stretch. So what we have here is this kind of business. So we have the muscle fiber here, the extrafusal muscle fibers. And then we have, some sensory neuron here that's going to pick up some stimulus in the environment. And then that signal or that firing response is going to be passed in through the dorsal root into the spinal cord. It should look familiar from previous lectures. And through the dorsal root, it's going to synapse with an alpha motor neuron here, and then a signal is going to be sent out through this alpha motor neuron through the ventral root to the extrafusal muscle fiber, and that's going to allow the arm in this case to contract or pull away. Now notice here that nothing here in this pathway is coming across any neurons that are projecting into the brain. All of this is going in through the spinal cord and then out the spinal cord through the ventral root back to the muscle. So these are again more auto responses like that kneecap tendon tap. Of course, we also do have reflexes that do involve the brain. These are called polysynaptic reflexes because these involved more than one synapse along the pathway of information coming in and signals coming out. So in this case, we have an impulse traveling in through a sensory neuron. So some sense neuron has picked up some stimulus. That impulse will then travel in that sensory neuron to a motor neuron, and it does so via an interneuron throughout the pathway in and out. This also does involve the spinal cord as well. But along the way, there are several synapses, and this is going to allow the signal to come across neurons that do project into the brain. And so what this is going to do is this is going to allow some processing in the brain and can also allow potentially for inhibition from the brain. So the great example is one that I used very early on in the semester when I was discussing response to a very hot stimulus, like a hot coffee cup or something that might be burning your hand, but you don't want to drop it because it will make a huge mess. You're getting that monosynaptic response of you need to put it down, it's burning your hand, you need to put it down, it's burning your hand, but you're also getting this type of response as well, where you have this impulse of the hot stimulus traveling in through the sensory neuron. Then it's also, again, it's going in through the dorsal root, but then you have multi synapses along the way, so it's coming back out through the ventral root. It is going to that muscle fiber. And if that was the end of the story, then your arm would just pull back and you'd drop the coffee cup. But it isn't. You have these connections with, for example, here an inhibitory interneuron. So this interneuron right here, this is going to allow for projection into the brain. So this particular pathway is going to come across more than one synapse. So we're not just going to have simple responsive contraction in the muscle. This allows for synapse with other neurons that are going to protect the brain areas that are going to allow you to inhibit that action of dropping the coffee cup. Now, if that happens, if we do have polysynaptic movement and that we have multi-synapses with information coming in through sensory neurons, and then we have those multiple synapses, and some of which are synapses with neurons that project into the brain. What happens once that information actually gets into the brain? So let's look at first at where it's going in the brain. First, we have that primary motor cortex. This is where that initial information is going to come, or that initial signal is going to come. Then we also have the supplementary motor area as well, and we can see these diagrammed out, and also kind of what we think is happening in these particular areas. Again, the primary motor area is going to be that primary receiving area. This is where things are going to kind of get to first, and then from there, we need to figure out what we're going to do with all that information. So it's going to project more into the supplementary motor area. And from here, we're going to be able to kind of have a, kind of a pattern of firing through the brain. In the prefrontal cortex, we're going to have things that are more involved with making plans of movements. Then in the parietal area, we know that a lot of what happens in the parietal area is more locational information, and also with regard to location, how to move your body accordingly to get to a certain location. This is also going to involve perception in space of kind of where your limbs are. Um, so again, with this, we're going to have our primary receiving area and also the supplementary area. This is going to be more of kind of what do we do with this information that's coming into the primary cortex? How do we plan for our movements? Where are we in space and in time? And how do we plan those movements accordingly to achieve perhaps an intended goal? This is quite interesting if we take a look more at the primary motor cortex. So here we have a slice here that's turned sideways for us. And we can see the different areas along the primary motor cortex and where they tend to be active when we have movement and stimulation in different areas of our body. And this little guy here looks really strange, but he looks strange because this is really interesting how different areas of the motor cortex are allocated or respond to or become active in firing to movement and stimulation in different areas of our body. This is called somatotopic organization. And what's interesting about this is that some areas are overrepresented. That's why our guy looks so disproportionate. That's because different areas of our motor cortex are allocated or respond to stimulation and movement of different areas of our body. And some areas of our body take over a lot more of this motor cortex than maybe they should if we are if it was represented proportional to our body. Here we'll look closer at a couple of the areas of the brain that we've already mentioned that are active when we are performing movements, and in particular, when we think certain areas tend to be more active. So here we're looking at the supplementary motor area. We think that this plays a critical role in what we call behavioral sequences, so things that take multiple steps to complete. We can look to non-human studies a lot to help inform us about how these areas seem to contribute or become very active when we are involved in things like behavioral sequences. So, for example, your book mentions a study with non-human primates where they injected mucimol into the supplementary motor area. And what this really does is kind of deactivate this area. And previously, what they had done with these non-human primates, they had trained them on a behavioral sequence. So it's kind of like a three-part task where they had to push a lever and then turn the lever a certain direction and do something else to kind of receive a treat. So there's kind of a three-step process here. And what they found was that the animals readily learned this, but when they were injected with mucimol into the supplementary motor area, these animals, they could still reach for objects and make some particular movements in response to certain visual cues, but they weren't able to perform the previously learned behavioral sequence when this particular area was deactivated by the injection of mucimol. Another area of interest is the premotor cortex. And again, we look to some non-human primate research to help us understand better how this area is correlated to certain motor behaviors that we perform. We think that this area is very active when we are executing more complex movements, ones that are guided by the senses, for example, and also really contribute to our learned responses to what we call arbitrary stimuli. Arbitrary stimuli are basically anything that um, are meaningless on their own, but they've been paired with a certain required behavior, for example. So let's say you're driving on the road and you see traffic lights, right? And green means go, red means stop. Well, red and green on their own don't mean that. But in the context of driving, through basic reinforcement and punishment, we have learned green means go, red means stop, and we behave accordingly. But again, red and green are arbitrary. They have to be trained to mean go and stop. And in particular, if you inject mucimol into the premotor cortex, was done in a non-human primate study that is talked about in your text here, it really inhibits responding to arbitrary stimuli. In their task, they um, had signal lights that would be presented on a screen. And what they found was that if there was a light presented on the left-hand side of the screen, then the animal would reach to the left. And if it was presented on the right, the animal would reach to the right. They then trained them that, for example, a red light signaled that they should move to the left, and a green light presented on the screen mean that they should move to the right. And the animals readily learned that. But again, that those are arbitrary stimuli. They're having to be trained to know that red means move to the left, green means move to the right. Now, once they get that injection into their premotor cortex of mucimol, the animals could still reach to the lights that were presented on the left and on the right, but when presented with red and green, they didn't perform the movement to the left or movement to the right. So it really inhibited their learned responses to the arbitrary stimuli of red and green. Here, this is a table from your text. I'm just going to present this here. It would be good to take a quick look at this just to see, in particular, where these specific brain areas tend to be more active given the different movements that we are performing. So if you take a look here toward the bottom, we have, for example, the frontopolar cortex, posterior parietal cortex. These seem to be more involved in intent, so decision to move, for example. Here we'll take a look at three different areas or subcortical structures in the brain: the reticular formation, the cerebellum, and the basal ganglia. All three of these areas contain nuclei, both in the midbrain and in the hindbrain. We find heightened activity in these areas when we are performing both voluntary and involuntary movements. And these areas tend to get information or signals sent from neurons that are projecting in from the motor cortex. And then from here, these structures then convey those signals on to the spinal cord and to the nerves. First, we'll focus on the reticular formation. This area in particular seems to be very active during involuntary muscle control, or related to those intrafusal muscle fibers, or those inside muscle fibers, relating to the gamma motor system. So again, involuntary muscle movements. This area does seem to be also really in control of our muscle tone or our muscle contractions. Large nuclei from this particular area are found in the medulla pons and in the midbrain. Um, Areas of the medulla seem to be playing a large role in automatic responses like sneezing, coughing, things like that. We also think that they play a role in posture, so how we hold our body, how we hold ourselves, also how we locomote or get from one place to another, and also more specific upper body movements. We see more activity here when we are involved in some specialized upper body movements. Next, we have the cerebellum, this round section here for the base of the brain. We see a lot of activity here when we're looking particularly at two things. So the timing of our muscle movements, if you think about it, it's quite important that we time our muscle movements appropriately so we can perform certain sequences successfully. For example, if you think about how you may swing a bat, for example, you have a very time-sensitive window there to do that correctly. We would see a good bit of activity here in this area when you would be involved in an activity like that. Also, we see a fair bit of activity here when our muscles are performing actions independent of one another. So for example, just because my right arm's doing it doesn't mean my left has to. And if my limbs are performing independent actions, we think that that is highly correlated with activity in the cerebellum. And if we lesion this particular area, we can see things like limb rigidity, also weakness and decomposition of certain movements, so just an ability to perform certain movements altogether, and in particular issues with things like ballistic movements, as we call them. So, for example, the action of throwing a baseball would be highly impaired by lesioning the cerebellum. And next we look at the basal ganglia. This is the particular area that receives a lot of its input. So axons projecting into this particular area are coming from the primary motor cortex and also the primary somatosensory cortex, as well as the substantia nigra. So from here, then we have neurons that project out to the frontal, parietal, and temporal cortex. So neurons leaving this particular area are projecting into these three different areas of the cortex. This particular area is very important because we find that neurons in the basal ganglia tend to play a large role in, or in particular, damage or degradation of neurons in this particular area are correlated to diseases like Parkinson's disease. In particular, Parkinson's occurs when we have degeneration of dopamine cells. in the substantia nigra. So cells that release dopamine and receive dopamine, we see degradation or cell death in Parkinson's patients, again, of these neurons or these cells in the substantia nigra. What this does is impair movement initiation. So the start of a movement seems to be very impaired in Parkinson's patients. Also, Parkinson's patients tend to have a very difficult time with maintaining balance or regaining balance if they are kind of tapped to one side. They may have a really difficult time regaining and recentering themselves. Also, this particular area, we find degeneration of both GABA and acetylcholine neurons in Huntington's patients. With Huntington's, we see inability to control movements, so uncontrollable movement. This could be constant shaking, for example, and also the inability to cease motor movements. So, for example, if a Huntington's patient has been kind of walking across the floor, they may have a really difficult time stopping that movement of walking. Let's chat for another minute or so about Huntington's disease or Huntington's chorea. Huntington's disease, I'm assuming more of you all are a bit more familiar with Parkinson's disease, but Huntington's also occurs with damage or nerve cell death in the brain. And like Parkinson's, we see a lot of that damage happening in the basal ganglia in particular. Huntington's is a progressive disease, meaning that it only gets worse over time and it is incurable. And again, it is related to the breakdown of nerve cells in the brain. And one proposition as to why this happens is some mitochondrial dysfunction, in particular, that leads to what is called oxidative stress. Oxidative stress can leave neurons just a lot more vulnerable to an excitatory neurotransmitter, in particular glutamate. And if you all remember from our psychopharmacology chapter earlier in the semester, if a neuron is overly susceptible to something like glutamate, an excitatory neurotransmitter, then it can get too much of it. And that's why after an action potential, when something like glutamate is released, it needs to be cleared away. But if a neuron is more vulnerable to it, it may receive too much of it. And this can lead to kind of burnout of the neuron or exotoxicity, which basically just means that the neuron is getting way too much of the neurotransmitter, and it's going to break the nerve cell down. And again, with this particular disease, the basal ganglia is most affected. So if we look here at structural changes in a normal brain versus the brain of a Huntington's disease patient, what you see here are areas of the basal ganglia, which is around the ventricles. If you all remember the lateral ventricles on either side of the brain from chapter 2 early on in the semester, those are those areas that are filled with CSF or cerebral spinal fluid. Here you'll see enlarged ventricles in the Huntington's disease patients because the areas around the ventricles, basal ganglia, have atrophied or kind of withered away. Here we look at a real life slice. Here is a normal brain on the left here, and on the right here you see how much larger the ventricle spaces that were filled with cerebral spinal fluid are, and you see how the tissue in the areas surrounding these ventricles have really recessed in comparison to a normal brain. And again, this is really as a result of this nerve cell death in this area. And again, we think that this could be because of excess vulnerability to things like glutamate that are really hypersensitizing those neurons and basically just burning them out. Now, some more information about Huntington's. Huntington's affects 1 out of about every 10,000 people, so it's a fairly rare disease. It affects men and women equally. It also, in most all cases, is genetic. So someone that has Huntington's is much more likely to produce an offspring that also has Huntington's. Unfortunately, Huntington's is what is called autosomal dominant, meaning it's a dominant genetic trait. So everyone who has that gene will develop the disease. About 10% of cases in Huntington's are due to new mutations, but almost all of them, again, are genetic and autosomal passed down from a parent. Onset of this disease, so most people tend to be diagnosed with Huntington's in their 30s or 40s. Earlier onset is much more rare, but if someone younger gets diagnosed with Huntington's, it tends to progress much faster. So symptoms of Huntington's, since we're focused on movement in this chapter, and the area that's most effective with Huntington's is the basal ganglia, symptoms include uncontrollable movements. Some almost appear to be dance-like. This is where that term chorea comes in. Chorea is a term for dance. Also, folks can have emotional issues. These could be things like depression, anxiety, aggression, and it's hard to say if that's due to the actual nerve cell death or if that's due to the response to other symptoms caused by the nerve cell death. They can also develop things like inability to control certain things, so hypersexuality, just overall compulsion, other addictions like gambling, for example, alcoholism, and basic cognitive impairments, so things like memory loss, inhibition of action, having a very difficult time learning rules, for example. And also, these folks can have metabolic issues, so can have uncontrolled weight loss. And of course, as you might imagine, suicidal thoughts are very common with Huntington's disease patients. Early signs of Huntington's can be depression, irritability, small involuntary movements, so small movements that are happening that are involuntary, poor coordination, and also trouble learning or making decisions. Unfortunately, Huntington's doesn't have a drug treatment, so there is no drug that's been found to be successful at treating Huntington's. The best they can do is try to manage symptoms, which is, of course, not helpful to the disease progression, but basically you're just trying to make the patient as comfortable as possible. This is a fatal disease. Folks with Huntington's will eventually succumb to the disease or some side effect of the disease, usually within 10 to 30 years of onset of symptoms. Common deaths with Huntington's disease include things like pneumonia. So folks with Huntington's over time can become unable to clear their lungs properly, so they can die of pneumonia. Heart disease is a common one, and then also another common one is fall. Because this is highly correlated with movement, folks with this particular disease are way more susceptible to having falls. And these folks also, over time, can really lose their ability to swallow properly. So another common cause of death for Huntington's patients can actually be choking. And malnutrition can also be a common one. As I said earlier, people with Huntington's can suffer from metabolic issues, and sometimes because they have issues with swallowing, they tend not to eat very well. So they can die over time of just malnutrition. Now moving on to more complex motor behavior and away from the very sad story of Huntington's disease. Let's take a look at how we perform more complex motor behavior. Your text mentions neurons called mirror neurons. We call them mirror neurons because they seem to become more active when we are observing other individuals doing behaviors. So for example, if we are observing someone manipulating objects, for example, we see these type of neurons become more active. And mirror neurons seem to be more allocated to the ventral promotor cortex. So more activity in this particular area of the brain when we're observing someone performing some action. And this makes a lot of sense. These particular neurons seem to be connected with other mirror neurons in the parietal cortex, which again, the parietal cortex is more active when we are involved in movement and spatial things, spatial location of things. So it kind of makes sense that these mirror neurons would be connected with other mirror neurons in the parietal cortex. And they seem to be most active when the behavior is competently performed. So we see, for example, this is very interesting. If we watch two models, one model that's performing actions very well, so grasping and manipulating objects very, very well, they seem to be very knowledgeable about what they're doing, versus if we're viewing a model that's kind of figuring it out, they don't really know what they're doing. They're trying to try different things to see what makes what happen. We see more activity in our mirror neurons when we're observing the model that seems very competent versus the model that seems to be learning. And what's interesting about this is that, and then this is a kind of an interpretation that we make based off of this more activity when we're viewing a more competent model. We think that it might suggest that this particular mirror neuron system might be trying to help us interpret intentionality. So if we're viewing a model that seems more competent, it seems as though their movements are more intentional, that they somehow know what comes next. In the movement A is intentional because it leads to movement B and then movement C. Whereas watching a model that's kind of doing things more clunkily, it doesn't really seem like there's real intent there because they don't seem knowledgeable. Quite active during pointing and reaching behavior. So again, more locational and spatial information. We see a good bit of activity in the parietal region when performing movements that are related to where something is in space. The last thing that we'll discuss for this chapter are a couple of issues with movement. So praxias. The first one we'll look at is limb apraxia. So this is when we have movement of the wrong limb, or incorrect movement of the correct part of a limb. Or we can also have an issue here with correct movements, but in the incorrect sequence. Then we have something called constructional apraxia. So this is issues more of how to draw pictures or putting objects together from different elements. So for example, blocks. If you're supposed to build a particular object and you have the different elements or the different blocks, you may not be able to put the blocks together appropriately to form the object that you're trying to build. This can affect our ability to perceive and also imagine geometrical relationships, so how things relate to each other in space geometrically, like, for example, corner to corner. So here, if we take a look at the brain, we have the left parietal lobe. And again, parietal lobe is really um, involved in where things are in space, locational information, and such. So with the left parietal lobe, if we have lesions here in this particular area, this can be correlated with limb apraxia, whereas lesions to the right parietal lobe are more associated with constructional apraxia. And here at the bottom, we have drawings. Um, the top one here is a model drawing, looks very nice. But then here you have the patient copy. So this is where you're told to copy the model drawing here. And what you see is they do pretty well, but you see issues with the corners being drawn appropriately and the space between the corners drawn appropriately. Just issues with constructing something or drawing something to match a model drawing. So that will be all for our lecture on Chapter 8. As always, make sure to go through this chapter material carefully, and I'll be back with you next week with a new content folder. Have a great week.